Hello, so today I'm going to be discussing the process of meiosis and why it's incredibly important when it comes to sustaining life. So first of all, meiosis is really important in sexual reproduction, which is why it sustains life. And what's important about it is that it reduces the number of chromosomes within a cell basically in half. So it turns a diploid cell, which is created in the first part of meiosis, and turns it into a haploid. So this is rather important because it prevents basically the child of the two parents who are providing the DNA here from having twice the amount of chromosomes as the parents, which would definitely be detrimental to the child's growth, as chromosomes have to stay at 23 pairs. It's pretty important. So meiosis has a pretty similar interface when it comes to mitosis. Same G1, same S, same G2 phase. I'll go over a brief refresher. So the G1 phase, it basically builds most of the proteins, carbs, and lipids that the cell is going to need to function. It's going to do its primary functions, of course, through just normal growth. In the S phase, the DNA is going to be replicated. Histone proteins are going to be synthesized to prepare for cell division. And then G2 phase, the cell is going to make sure it's ready for cell division, or ready for the cell division to take place. And it's, of course, the shortest between G1 and S. So now we actually move on to the process of meiosis, which is what we want to focus about in this video. So now when it comes to meiosis, there's two sets of cell division, like I mentioned earlier. So I'm going to be referring to them as, of course, 1 and 2 when I talk about them. So if you hear me say prophase 1 and prophase 2, prophase 1 is, of course, the first cell division happening in meiosis. Prophase 2 is the second division, and, of course, so on and so forth for metaphase, anaphase, telophase. Simple stuff. So once interphase is done and we can actually move on to cell division, prophase 1 is going to start. And the chromosomes are going to, of course, begin to condense. But because there is two sets of chromosomes coming from two different parents, there's going to be basically double the amount of chromosomes compared to mitosis. So, for example, compared to the mitosis video, which you can also see on this channel, we did the chromosomes looking like this, where we have our centromere at the center connecting them and, the two pairs. However, when it comes to meiosis, we have two homologous chromosomes here, because of course they're from two different parents. So say these are our two homologous chromosomes, right? We have parent A, or our parent A, parent B. So let me try to cross that a little better. So when they combine up and they condense, they're going to pair up as what we know as tetrads. So watch what happens here. I'll try to get my fingers out of the way, but look in the center. They're going to cross over and connect. So this crossing over is super important. As you can see, our pink highlighter crossed over with a pencil, and then our pen crossed over with a yellow highlighter down on the bottom, if you can see that there. So that's super important because what's happening there is it's allowing for differences and changes to be made in the child, or the child's DNA to be created. It's basically combining the two different DNA strands and almost shuffling them together, per se. And now that they're condensed and all bundled up together, we can now move on to metaphase 1. So in metaphase 1, these tetrads of chromosomes are going to line up along the metaphase plate, or at the center of the cell, and are going to be connected to each end of the pole with the spindle fibers, almost just like in mitosis again, which is almost going to look like this with our, if our chromosomes were in the center, these will be our spindle fibers, each connected to our different pairs of chromosomes. Now, they're not going to basically, pull all of them apart. They're going to split them into pairs now, because they weren't tetrads, so they're going to now split them into pairs. So we'll move now on to anaphase 1, which is, of course, that splitting. So the spindle fibers are going to pull the chromosomes to each part of the cell, but what needs to make sure it's important is that when they're pulled together, or pulled apart, for example, let's get our pencils again. When they're pulled apart, oh, I dropped one. Give me 1 second. And they're pulled apart, the sister chromatids, even though they've been, maybe shuffled around a bit, we changed the pens and such, they've been moved. But the sister chromatids don't separate in anaphase 1. Very important. The sister chromatids are going to stay connected at the centromere and be pulled apart. All right? So that's what's happening during anaphase 1. And in telophase 1, the chromosomes are going to decondense, and the nuclear envelope is going to form around each, just like in mitosis. Cytokinesis is then going to occur afterwards in animal and fungi cells. It's going to form like a pinch and kind of break them off, and then in plant cells, it's going to form a basically a plate of a plate, which is a cell plate of vesicles, which will then form the cell wall of each different cell. Now, when it comes to sorry, I lost myself here. Sorry about that. So now that we have our cells that have been split after telophase 1, so now we have our two diploid cells. So they have double the amount of chromosomes compared to the parent cells. So what do we need to do now? We need to split those cells again. So now we're going to move on to our second split and turn those diploid cells into haploid cells. So we have basically four end cells at the end of meiosis. So now prophase 2. So we'll be focusing on our two different cells, but for simplicity's sake, we'll just work with just focusing on one. So with prophase 2, the spindle fibers are going to reform again, and the chromosomes are going to recondense just like they did in prophase 1 and cross over each other once more. So remember, now we only have, just double our set, but they're going to cross over again. Now, I know it might be a little confusing, of course. However, they're not identical, or rather what's important. So in our prophase, they're condensed, and in metaphase, back to prophase. My apologies, I got a little lost there. So in prophase 2, the spindle fibers are reformed and the chromosomes are going to recondense like they did in prophase 1 and are going to cross over again. So there's going to be another basically kind of switch or crossover of the chromosomes, which is basically going to change each different haploid cell into different pairs of chromosomes. So they're no longer going to be identical across the board. Each different cell will now be identical, or each pair of chromosomes will be identical. Now in metaphase 2, the chromosomes are going to be once again aligned onto the cell plate. Now this is for both cells. We're kind of working at the same time. And the pairs of chromosomes are going to be attached to the spindle fibers, just like metaphase 1. Unlike metaphase 1, though, there's going to be just pairs of chromosomes, unlike the tetrads we were looking at earlier. So unlike the duplicate pairs of chromosomes kind of crossed together again, we're just going to have just normal sister chromatids, so just two pairs. Or just a single pair, rather. And then in anaphase 1, the sister chromatids are going to be, once again, separated by the spindle fibers, or rather, the sister chromatids are going to be separated by the spindle fibers, unlike metaphase 1, in which they say connected at the centromere, and we just split the tetrad. Now we're actually splitting them at the centromere and pulling them to the opposite end of the cell. This is very similar to the mitosis anaphase. So if you want to look at something that's very similar, that's a good representation. And then finally, in telophase 2, which is our last part of meiosis, the chromosomes are going to be once again decondensed, and a nuclear envelope is going to form, and then cytokinesis is going to happen, which is going to split those two diploid cells finally into those 4 haploid cells we're looking at. And then they're super unique, like I mentioned earlier. At least each one is unique to each other due to the crossing over of the chromosomes that happened when we had our tetrads, and due to that separation of the cells when they got pulled apart during anaphase, that also kind of shuffled them around as well. So this is, like I said, this is super important when it comes to sustaining life, with keeping new kids passed along with proper amounts of genes, and it makes sure that there's change within each one of them so there's not problems with genetic defects. All right, so that's going to be my video on meiosis and the processes associated with it. Thank you for watching, and have a great day. Bye-bye.